Welcome to lecture 12 in biopharmaceutics and pharmacokinetics, and today's topic is report format of bioequivalence studies, and it is the last topic in the chapter of bioavailability and bioequivalence. So today's lecture we're going to discuss about all those parts which are part of the format of the report for the bioavailability studies, which is supposed to be submitted to the respective drug regulatory authority. So the important parts include title page, protocols and approvals, clinical study, assay methodology and approval, pharmacokinetic parameters and tests found out during the study, statistical analysis and in vitro testing. As we initially discussed that before carrying out the bioequivalence of or bioavailability studies, in vitro testing is performed for the products which are being compared, and then the batch record. So important information which is part of the title page is first of all the study title or the title for the study which was carried out and during which the bioequivalence or bioavailability was determined. And then other information would be which would be part of the title page would be the name of the sponsor, whoever is financially sponsoring the study. That could be any organization or a company, name and address of the clinical laboratory where the study is being conducted, name of principal investigator and name of chemical investigator. Sometimes both investigators are the same. However, if both are different, then both names must be mentioned. And name of analytical laboratory where the analysis will be performed. Dates of clinical study along with start and completion. Signature of principal investigator along with the date mentioned there. Signature of clinical investigator along with the date. So the second part is the protocols and approvals. So this report is not just a single page report. It has so many parts which are attached with the report, including protocol. Whatever protocol is being followed for the study, that must be attached along with the report. And letter of acceptance of protocol from respective drug regulatory authority. They usually have a set format for that purpose, and so usually the protocol which is adopted by the study team is the one which is approved by that respective authority. So there must be a letter of acceptance attached. Then informed consent form. As we know that during the clinical trials or clinical studies, volunteers are included. Volunteers can be healthy volunteers or the patients, but whatever is the case, informed consent is very important. So there is a specialized form for that. So that form also needs to be attached. Letter of approval of institutional review board. One approval is required from the respective drug regulatory authority. One is required from that institution as well where the study is being carried out, and then the member of list of the members of the institutional review board is also required. Then clinical study, as we know that bioavailability studies or bioequivalence studies are basically the clinical trials. So these studies are conducted according to a set protocol, and then the summary is provided for the study that whatever steps were involved in that. And usually clinical trial has two proportions. One is the one which is dealing with volunteers inclusion exclusion criteria or their blood sampling is involved, or the physical fitness tests before their inclusion in the study is involved. So another detail which is usually have to be given is the demographic characteristics of the subjects. Usually this information includes the gender, age, or any ethnic characteristics associated to them. Various biochemical tests are usually involved, which have to be provided. So all this information is attached along with the report. And subject's assignment in the study, usually the subject assignment is the drug, whatever drug is being administered to them, and what criteria has been told to them to follow. Like how many days before the clinical trial they are not supposed to take any other drug, or during the washout period, what intake they are allowed or not. And similarly, what is the standard meals set for them during the clinical trial. So all the information is given. Uh, any deviation from the protocols is, if happened, that details should be provided also. Details of the clinical study must be mentioned. After conduction of the clinical trial, then the analysis phase comes, and analysis purpose, whatever assay has been adopted, its methodology, it needs to be validated. As we know that in case of for the analysis purpose, 60% of drug analysis is performed with the help of high performance liquid chromatography. So for example, the method or procedure of chromatography which is being followed, that should, its summary should be provided, and whatever method of validation was followed. And usually the method of validation for the chromatographic procedure is to provide the data on linearity, accuracy, and precision of the method, and also the figures of the standard curves, which we already know that are obtained by plotting the concentrations of the drugs obtained versus time profiles. And then chromatograms of standard and quality control samples must be provided, you can say, as an evidence, or to show that the method followed was accurate. And chromatograms are usually an indication that proper methodology was followed. Pharmacokinetic parameters and tests. Um, During this study, as it is a pharmacokinetic study, so various pharmacokinetic parameters, including area under the curve, which is a very important estimate of bioavailability studies, is performed, are determined. And so their determination involves various calculations. So whatever equations have been adopted or software has been used, then for information regarding that is provided. Also the definition of various pharmacokinetic parameters glossary is provided. Statistical tests which were adopted for carrying out the various performing the significance of difference whenever the bioequivalence study was being performed between various products. So whatever test was, statistical test was used, those tests are also mentioned. And drug levels at each sampling time, and pharmacokinetic parameters, basically the concentrations. And then after that, the concentrations are plotted versus time, which are called the plasma concentration versus time profiles. And then with the help of these curves and also using the softwares, pharmacokinetic parameters are also determined. And so figures of mean plasma concentration time profile and figures of individual subject plasma concentration time profile. Now, nature is the same, but both things are different from each other in a way that in case of figures of mean plasma concentration time profiles, these profiles are actually displaying the mean concentrations, or you can say the mean value for the concentrations at a certain time point for all the volunteers which were part of the study, while the figures of individual subject or plasma concentration time profile are basically displaying the individual concentration at a certain time point. It is not mean value, it is not displaying or it is not depiction of all the concentrations of all the volunteers at a certain specific time point. So both the figures are provided, and then figures of the mean cumulative urinary excretion and figures of individual subject cumulative urinary excretion is the same thing. But the point is, if the data has been the data which was collected or the sampling which was performed, if it is not the blood sampling and it was urine sampling, then the data or the figures would be of the mean cumulative urinary excretion and the individual subjects cumulative urinary excretion. It won't be the mean plasma concentration time profile. Statistical analysis is the continuation of the same thing. Summary of statistical significance and parameters. Various tests, as we discussed earlier, that various tests are involved in order to perform or in order to find out if any statistical significance is there for the differences which the formulations hold towards each other. So suitable statistical tests need to be adopted, which can be of various kinds. With, analysis of variance and t-tests are some few very important tests, popular tests which are adopted. So the details of any such tests which have been adopted to perform the statistical analysis needs to be provided, and assessment of sequence, period, and treatments. Actually, as we discussed earlier, that there are various designs towards the study, clinical study. This could be parallel design, this could be crossover, this could be two-way crossover or the three-way crossover, depending upon the formulations involved, number of formulations being compared involved. So if in case of crossover design, what happens that number of treatments or the sequences are greater than one. So if such a pattern has been adopted, so how their suitability was assessed, if there was any protocol or methodology was there, the information also needs to be provided. There are certain appendices attached with the report, which provide the schedule for randomization, which is performed during the clinical trials, and if any identification codes were assigned, so the list is also provided, and analytical raw data, medical records. Analytical raw data is the one which is initially obtained, and it is basically the chromatograms area, and with the help of a standard usage during the analysis, then this area is used for determination of concentration. So that initial chromatogram areas are the raw data, which is the analytical raw data before it is converted to the respective concentrations. So that is provided, and the medical record and clinical reports basically of the volunteers which are part of the study, as I initially described, that these volunteers are made part of the study only after the performing certain physical tests, and they should be physically fit in order to be part of the study. So all those reports must be part of this report, which is being submitted to Drug Regulatory Authority. Clinical facilities description and analytical facilities description is also part of the report and attached appendices. In vitro testing is performed before carrying out the in vivo part of bioavailability studies, and dissolution testings are involved in that. So dissolution assay methodology is details needs to be provided. Content uniformity testing is In the last, it is also important to provide information regarding batch size of the formulation which was developed and was being compared with the standard reference. What was the size of that batch, and what was uh, and all the other information related to that batch, its record, that must be provided, and that should be attached along with the report as well.